Hello everyone, I'm Rania and today I'll be talking about Rosalind Franklin's investigation of the DNA structure, which produced an image we now know as Photo 51. I've created a model as you can see here, which is what I'll be talking about today, but first I'll be beginning by talking about the history behind this photo. To begin with, this pattern of DNA was obtained by Rosalind Franklin and Raymond Gosling, who was her graduate student, in 1953. This photo, called Photo 51, is one of the most famous X-ray photographs because of the fact that it was clearer than others. The reason it was so clear is because water was included in the DNA sample, and this helped us show the distinct helical structure you see in the image. Photo 51 is of the hydrated B-form DNA, instead of the A-form DNA, which was too dry and didn't show the helical structure. Maurice Wilkins was also involved in this investigation, since both him and Rosalind Franklin were investigating the structure of DNA. Unfortunately, she didn't want to publish her findings until after there was strong evidence that its structure was definitely helical, but meanwhile Francis Crick and James Watson used her findings to build a model of the DNA structure. It is said that Maurice Wilkins showed Photo 51 to Watson, who then used it as he wished. Essentially, she didn't receive any proper recognition for her work. So, now that we understand a bit about the history behind this image, I'll be talking about the method used to help produce it. As mentioned earlier, Photo 51 was obtained using X-ray diffraction. When X-rays are directed at a material, most of the rays pass through the material, while some get scattered by particles. Because of the wavelength of the X-rays, they become sensitive to diffraction by particles in the biological molecules, like DNA. Now, in crystals, particles are arranged in regular repeating patterns, meaning that diffraction would also occur regularly. Now, although DNA can't be crystallized, Rosalind Franklin arranged molecules in her sample so that a diffraction pattern could be obtained, and so that the scattering wouldn't be random. And so, an X-ray detector was placed close to her sample, and it collected the scattered rays, and the sample was rotated in 3 dimensions to obtain the pattern of the rays scattering. Then, an X-ray film was used to capture clear images of the patterns. This is the method that Rosalind Franklin used, also known as X-ray diffraction. So now I'll be talking about the model I made and how it helped Franklin make deductions about the DNA structure. So to begin with, the X in the model indicated that the DNA was helical in shape. In addition, because the pattern was so regular, it was deduced that the dimension of the helix must also remain constant. For example, the diameter of the helix stays the same, and we now know this to be true. The next deduction made about the DNA structure was that the angle of the cross shape showed the helix's pitch, or its degree of rise that it makes with the horizontal axis. So, if we make our X, and this is our horizontal axis, then this angle right here corresponds to this one right here, which is the helix's pitch. Another deduction made was that the distance between horizontal bars was 34 angstroms, or 3.4 nanometers, and this distance corresponded to helical turns. That means that this distance right here, which is 1 helical turn, would also be 34 angstroms, or 3.4 nanometers. The final deduction made was that the distance between each of the horizontal bars was 3.4 angstroms, or 0.34 nanometers, and this must correspond to the distance between base pairs. And since they were able to deduce that the height of a helical repeat is 34 angstroms, and the distance between adjacent base pairs is 3.4 angstroms, they were able to calculate that there must be 10 nucleotides per each helical repeat. In conclusion, all the deductions that I talked about today were very important in the discovery of the structure of DNA, and hence have acted as a starting point for many great discoveries, and many discoveries that are yet to come. Thank you for watching, and I hope you learned more about Rosalind Franklin's investigation of the structure of DNA.